My name is Jordan Tapley, and I'm going to be talking today about how repitching and acid washing of brewery yeast strains potentially decreases the environmental stressor response ability to cause a decline in foam head stability of beer products. The goal of my experimental study this summer it was to exhibit a decrease in the ability of brewery yeast strains to respond to environmental stress responses. And specifically in this case, I'm looking at pH change, sudden drops in pH, and also repitching of the yeast as well, leading to an overall decline in foam head stability in your beer. In this presentation, I will go through my background and some important defined terminology that I'll be using throughout the presentation, then my hypothesis, my research design, experiments, and methodology, and then my results, my projected results and analysis, and then the conclusion, kind of bringing everything together in the end and how everything is significant in the big picture. So the yeast that I was using it's called Saccharomyces cerevisiae. And this is a very common yeast strain used in a lot of different industries, from baking, winemaking, and in this case, beer brewing. And so its main role in these processes, it's what everyone uses it for, is its ability of fermentation. And fermentation is the production of ethanol or alcohol and carbon dioxide from glucose in its environment, and also in an anaerobic environment, meaning oxygen is not needed. It can be there, but it doesn't need to be there. In the brewing process, just kind of an overview of the beer brewing process, it's divided into 4 primary stages. The first stage is your maltings and brewhouse stage. Then you have your cellar stage, then filtration, and then you have packaging and distribution. And the 2 stages that I'm mainly focused on in this study that I'm more concerned about are the first 2 stages, and I'll tell you a little bit more about those. So your first stage, maltings in the brewhouse, this is where your barley or whatever grain you're using to make your beer is converted to a malt. And that's a substance that provides flavor, color, and the fermentable sugars that your yeast is going to need later on. And also the malt is mixed with hops and different ingredients to produce a liquid solution called wort. And wort is the main source of nutrients for the yeast to grow and proliferate during fermentation. And wort also contains proteins, which I'm concerned about as well, because it also contains something called lipid transfer protein 1, or as I'll refer to it as LTP1, and I'll talk about a little more about that in a bit. So you have your cellar stage, which is your second main stage of the brewing process, where your yeast are converted your yeast convert the fermentable sugars in your wort to ethanol and carbon dioxide. And this is also where flavors are developed, specific color is achieved, so a darker versus a lighter brew, depending on the caramelization. And also this is where your yeast go through something called yeast handling. And it's either filtered out, called cropping, to be reused, or as I refer to it, as repitched, or it's discarded because it's been repitched too many times. And then what I'm also looking at is the environmental stress during this yeast handling process. And it's during the cropping and repitching step where the yeast can undergo significant environmental stresses due to changes the environment is undergoing. And mainly what I'm looking at is acid washing, which is where the yeast that are set to be reused or repitched, they're put into highly acidic environments, so the pH is lowered to about 2.0, and in very cold conditions as well, so about 4 degrees Celsius, which is right above freezing. They're put there for a few hours to kill off all the bacteria that might be there, and then they raise it back to pH, which is around 4.7, and then back to regular temperature, they wash it, and then they reuse it. And then the significance of this in the brewing industry is that I'm looking at something that it affects called foam head stability of the beer. And one very strong focus of breweries is customer satisfaction and quality management. And it's because customers look for certain aesthetic qualities in beer, color, flavor, and what I'm looking at is foam head stability. Beer head foam plays a major role in key aspects of beer consumption, both physically and chemically. Physically, it's an aesthetic quality. A lot of customers desire a certain amount of foam in their beer, and that it stays there for a prolonged amount of time. And also, the stability of the foam, it enhances the flavor. The flavor molecules are more able to travel through your olfactory senses and kind of causes just overall enhancement of the flavor. And then, like I mentioned before, LTP1. This is a protein that is responsible for this foam head stability. And LTP1 is actually degraded by an enzyme that can be found in the beer called proteinase A, or PRA, which is encoded by a gene in the yeast called PEP4. PRA is activated at about a pH of 2 to 2.5, which just happens to be the pH that we lower it to during acid washing. So as the yeast become and also as the yeast become repitched or stressed during this, so as it becomes repitched over time, the PEP4 will start producing more PRA. So as it's repitched, more PEP4 will start producing more PRA, and then when it's acid washed, more of that PRA will be activated. So that LTP1 gets degraded, and it's just an overall process like that, which leads me into my hypothesis. So what I expect that will happen is, first of all, I'm looking for a relationship between PRA and LTP1, and if it's present, what environmental stresses are responsible for this? And I'm looking again at pH, acid washing, and repitching, and the fact that it's been repitched versus not. And so, like I said before, I think that repitching the yeast will cause PEP4 to express more of that protein, of that PRA. And then when you acid wash it, more of the PRA that's present will be activated, more LTP1 will be degraded, and your foam head stability will be degraded as well over time, degrading your beer quality. So I performed two main experiments in this study. I'll go through each of them individually. The first one is a proteinase fluorescence assay. And this is where I looked at the amount of activated proteinase A, PRA, in the final beer product. And so my question here was, does, number one, a change in pH affect activated PRA? And does repitching affect the amount of activated PRA? So I had two parts that I kind of combined for controls and treatments. So I put all of my strains that I used at 3 different pHs. I had the initial pH that I kept a group at, which is around 4.7, depending on the strain you're using, 4.5 to 4.7. I left it alone. And then the other, I put these in two other pH groups, 3.5 and 2.0, and 2.0 is an acid washing pH. And for part two, I used 3 different lab strains, or I'm sorry, 3 different yeast strains. One strain kind of uses like a control. One of my controls is a lab strain, and it's not used in a brewery. It's just kind of for reference, and also to see maybe how domestication versus lab strains might affect it for maybe later studies. But we'll get to that later. But another control that I had was a new brewery strain. The other 2 strains I had were brewery strains used in an actual brewery. One of my strains was one that's never been used before, never been pitched. And then my treatment, my experimental strain, was one that had been repitched multiple times. And that way I had 3 different lab strains, 3 different yeast strains, and 3 different pHs. And then I used a proteinase assay kit to quantify the amount of activated PRA using fluorescence testing. And my projected results here, so as you can see, what I expect to happen is that the lower the pH, the more fluorescence you're going to have, meaning that the lower the pH, the more proteinase A is activated. And then also at repitching, so for strains that have been repitched multiple